Quality control is an essential step in assuring that reliable and accurate results are produced in the laboratory. This is done by testing quality control materials. Quality control materials, or QC materials, are used in the laboratory in a daily basis. So these are specimen or solutions which are analyzed solely for the quality control purposes of the laboratory. They are specially prepared for this purpose. They may also be available commercially. Quality control materials have known values. So alam natin kung anong value nila. So for example, we have a quality control for cholesterol. That quality control material will have a specific value. So when we use it to check for accuracy, dapat yung result na yon, the known value that that quality control has should be reflected. Now, quality control materials are also known as control product or control solutions. Now, given that quality control materials have a very important role in determining if the test procedures are accurate, there are ideal characteristics that quality control materials should have. First, it should closely resemble the real specimen. What does this mean? It means that it should behave like the real specimen. Remember, quality control are specimens or solutions that were produced in the laboratory to be used for testing like a specimen. So that means they should be like a real specimen in physical characteristics or in chemical characteristics. Now, since in our laboratory we are testing people, human, then that means that it should reflect human specimen as close as possible. Why do we need them to behave like the real specimen? So that we can test them exactly the same manner as the patient specimen. Next is the availability in sufficient quantity for at least 1 year. We say that they should be available in a very long time para hindi tayo laging palit-palit ng control materials, because when we change control materials, there's a lot of adjustments done on the machines, on the reagents, and so on and so forth. So it's good that if we have a control material that can last us for a whole year. Now, given that we want this type of characteristic, it is also important then that they should be stable over a period of availability. So kung available siya for 1 year, then that means that it should be stable for 1 year. Yung hindi siya masisira within the year. There should be a long-term stability. But nowadays, there are a lot of quality control products that are stable for more than 2 years. They have 2 years expiration. The next one is that it should be available in convenient vial volumes and cost-effective. Cost-effective, of course, we know that it should not be too expensive or that expensive. When we say available in convenient vial volumes, that means that it should have a convenient packaging so that dispensing and storage would not be a problem. Next, vary minimally in concentration and composition from vial to vial. This means that there should be a low vial-to-vial variability. Kung ano yung characteristic ng isang vial, kapag nag-open ka ng isang vial, they should be mostly the same, or they should not differ at all. Next, it should span the clinically important range of the analyte's concentration. That means the levels of the controls should be appropriate for decision levels. It should reflect the clinical range. For example, we are testing for triglyceride. Sometimes a triglyceride can go above 200. If our quality control material does not have the ability to detect more than 200, then anong sense nun if we cannot check that we can test a specimen over 200? So that means it should be available in appropriate decision levels. And lastly, of course, no communicable diseases. Since we are trying to have control materials resembling human specimen, they recycle human specimen. And of course, when they do this, there might be diseases that accompanies them. So it's important that the control material being used does not contain any communicable diseases or is not infectious. So where do quality materials come from? Now, there are two sources for quality control materials. We have first the human serum. Now, usually, these are the homemade materials, the ones that the laboratory makes, yung inipon-ipon ng laboratory para makagawa ng quality control material. Now, this can be difficult to minimize infectious diseases since we don't screen every single serum with all the possible diseases. It's possible na meron palang serum doon that is positive for something that we do not know, and when we use it as a control, it's an agent for infection. They are also more susceptible for deterioration and contamination. So human serum have the possibility of losing its potency more faster than the other source. Also, you need to have separate controls for the specific analytes. Since iipunin mo lang to, for example, you need to have a separate control pool for blood sugar, a separate control pool for cholesterol, a separate control pool for triglycerides. So that's a lot of control pools that you would have to collect. Why? Because you would need a specific value only. So dapat mag-ipon ka muna ng serum na may pare-parehong sugar, mag-ipon ka ng serum na may pare-parehong cholesterol levels, and that's what you make as your quality control materials. The second one is animal-based serum. The most common animal serum is the bovine-based serum. Now, this is lower in price than the human-based. Its stability is the same with the human-based, but it is inappropriate for immunochemistry and dye binding procedures. So, which one is preferred? Remember, for one characteristic for quality control materials is that it should resemble the specimen being tested. Now, sino pang mas lalapit sa specimen na tinetest natin than the human serum? Of course, a human serum control material is very similar to what we are testing in the labs because we are humans, right? But because of certain potential biohazard risks today, the bovine-based control products have become more popular. So what are the different types of quality control materials? We have two. We have commercial and homemade. When we say commercial, these are the ones that you can buy through the medical representatives. These are usually obtained from manufacturers who specialize in the production of control materials, and often they are provided by the same companies who would be selling the reagents and the kits from your machine. Now, under commercial, we have two, assayed and unassayed. When we say assayed, this is more expensive than the unassayed. Why is it more expensive? Because it already has an established control value. This is most useful for infrequent tests, yung mga tests na hindi mo laging ginagawa. Unassayed, on the other hand, will only have a target value. So that means you would have to test it and then the actual results should fall near the target value. If that is the case, that means you should perform data analysis. That's why this is appropriate for frequent assays, because when we have a lot of tests, a statistical data can be quickly accumulated. Unassayed commercial quality control materials are the ones that are mostly used. Now, the second type of QC is homemade. We have introduced this earlier, so this is the in-house quality control material. These are the pooled sera collected in the laboratory, and they are preserved in small quantities, as you can see from the picture behind this. They are preserved in small quantities for daily use. There are different variations when it comes to the quality control materials. They can come in lyophilized form or they can come in liquid form. When we say lyophilized, these are the freeze-dried quality control materials that come in powder form, as you can see in the picture. So before using it, you have to reconstitute it with water or specialized diluent. We say reconstitute when we add a diluent to a powder form like this one before using. Now, there are standardized steps in reconstitution. The liquid form, on the other hand, does not require reconstitution, and that makes it more expensive. Why? Because it is already ready to use. But one downside of it is that it contains additives or preservatives that may introduce error due to matrix problems. What is matrix? A quality control matrix is the base added to the control material. For example, the spiking materials or the preservatives. Now, ideally, there should be the same matrix as the specimen so that it can behave as the real specimen. Let's now differentiate a control versus a calibrator. Earlier, we've all been talking about a quality control material, so that's the control. But there's also this one solution that we use in the laboratory, and we call it as a calibrator. Now, the calibrator is different from the control. A control is a specimen or a solution which is analyzed solely for quality control purposes. It is not for calibration. A calibrator, on the other hand, has known concentrations of substances or analytes being measured. Now, this kind of has the same meaning as a control. Remember, the control also has known values. The calibrator also has known values, but there's a difference. A calibrator is used to adjust the instrument. It is used to adjust the kit, or it is used to adjust the test system in order to standardize the assay. So a calibrator is a substance with a specific concentration that gives the reference point for the instrument to adjust to. So it is used to set the machine or to calibrate the measuring of the machine. Sometimes a calibrator is also called a standard. So again, a calibrator is not a control. So before you use the machine, you should calibrate the machine first, because we use the calibrator to reference the machine, to standardize what the machine knows. So for example, you use the calibrator with the known concentrations. Tuturuan niya yung machine na, machine, this is what 100 milligram per deciliter is, this is what 200 milligram per deciliter is, and so on and so forth. So after you calibrate, after you adjust the machine to the reference values, then you can run a control. Now, a control, as mentioned earlier, is to test for the accuracy. Kung tama ba yung mararun natin. Correct? So when you run the control and it's calibrated properly, it will give the correct result. A 100 would be a 100, a 200 would be a 200. Now the problem happens is, for example, the machine is not calibrated properly. So that means hindi niya alam yung reference values. It might mean that yung 100, hindi niya alam na 100, baka yung 100, 200 para sa kanya, yung 200, 300 para sa kanya. So when you control, even if you have a known value, kapag nirun mo yung control, since hindi niya alam yung totoong value, for example, your control is 100, and you run it in a machine that is not calibrated, hindi niya ibibigay yung 100 na value ng control. Ibibigay niya yung alam niyang value ng 100, which is 200. Kasi ay alam niya yung 100, 200. So that means it's not calibrated properly. So kahit na anong gawin mong run ng control, kung hindi naka-calibrate yung machine, hindi niya makukuha yung totoong value ng control. Let's give another example. So for example, in our class, the calibrators would be the teachers. So the teachers will teach the students, so the machines are the students. The calibrators will be the reference point. We will teach you what hematology is, what chemistry is, what clinical microscopy is. We will give you all these references. We will teach you everything. We will tell you what you should set yourself into. And now, to check if you are calibrated properly, we will now give you quizzes. Now, the quizzes class would be the control running. Now, if in the control running in the quizzes, tama yung mga sagot ninyo, that means you were calibrated properly. But for example, if the calibrator did not teach you correctly, hindi tama yung mga tinuro niya, when we go for an exam, when we test for the control, yung mga sasagutin ng student, mali din. Why? Because the calibrator is wrong. Correct? So I hope you kind of understand it, that the control is not a calibrator and the calibrator is not a control. These are two different things. You should calibrate first before performing the control. Kapag pasok ang calibration, dun ka palang pwedeng mag-run ng control. I hope that helped you understand quality control materials. If yes, click like and subscribe. Thank you very much for listening.